Hey, welcome to section 2.4's video. So we're going to start this video off with a review question. So the question is, an epidemic is which of the following? So A, the continuous but low-level presence of a disease in a community. B, an increase in the number of cases of a disease in a community. C, caused only by airborne viruses. And D, a disease that spreads rapidly worldwide. Okay, so take a second and pause the video and see if you can find the answer. Okay, so let's go through the options. So the answer for this question is B. An epidemic is an increase in the number of cases of a disease in a community. Okay, so let's look at the other answers. So there's one here that's not like the rest. It's not correct, and that would be C. So an epidemic is not caused only by airborne viruses. Endemics and pandemics aren't either. They can be caused by fecal oral route and any of the other routes of transmission that we talked about previously in this chapter. Okay, so A is the definition of an endemic, and D is the definition of a pandemic. So this is a good example of the type of question that would be asked about these three definitions on the exam. So this is a good one to know. Okay, so section 2.4 is disease portals and biosafety. Okay, so we're going to talk about how disease enters the body and how we take precautions to prevent that entry. Okay, so our section objectives are we're going to talk about how infectious agents get into the body. We're going to be able to describe the various portals of entry and exit for microbial pathogens, and we're going to talk about a concept called biosafety and biocontainment and how that applies to us in the lab. Okay, so I'm actually going to exit out of my little picture so we can see everything. There's a lot of writing on these slides. Okay, so the first thing we're going to talk about is how do infectious agents gain access to our bodies and to cause disease? Okay, so we have a lot of routes of entry and exit, so different portals that a pathogenic microbe can gain entry to your body. Okay, so the first one is one that we've talked quite a bit about. It's the fecal oral route. Okay, so you ingest either some contaminated food or contaminated water that has feces in or on it. Okay, so it's now in your body. So it's going to enter your GI tract through your stomach and then enter your intestines. Okay, between the small and large intestine alone, there is a lot of surface area. So the pathogens that gain entry to your body via the fecal oral route are going to have a lot of places in this surface area that they can then use to transfer themselves to different parts of the body. Okay, the next portal of entry is your skin. So your skin is actually the first line of defense for your immune system. And as long as it's intact, it prevents infectious agents from gaining access to your body. So it's when you get cuts and nicks and any wounds that open your skin up, this is when you can be vulnerable to a infectious agent gaining entry through your skin cells. Okay, so it'll come through the epithelium. The third route of entry would be your respiratory system. Okay, the portal is the mucosa that is part of this respiratory tract. So this is going to be the mucosa in the back of your nose, in your lungs, and so forth. Okay, mostly this is going to deal with your airborne pathogens that you inhale. Okay, the next one is the urogenital route. Okay, this is when pathogens can enter directly through contact with the mucosa of the genitals, okay, either through skin to skin contact or through sexual transmission. Okay, the next is the parenteral route, and if you remember, parenteral route is how you can gain an infectious agent either by an insect bite or from a needle stick. Okay, so it's going to utilize breaks in the skin. Okay, one of the important things here, we've talked about the insect bites, but a lot of parenteral disease is actually caused by contaminated needles. So in the the drug junkie community, they're going to swap contaminated needles back and forth and use them to get their high. And this is a very extremely efficient way to spread disease in that community. Okay, the last one we're going to talk about is entry via the eye. So pathogens can enter through your eye using the conjunctiva. This is the membrane lining around your eye. Okay, so this could either be by respiratory droplets, so somebody sneezes in your face and it enters through your eye membrane that way, or it could be something it's as simple as not washing your hands and then touching your eyes with your hands that picked up the germs and letting it enter that way. Okay, a lot of times entry via the eye is through indirect transmission using fomites. So you go and you grab the door handle that somebody that had wiped their nose on their hand right before you who had used it, and then you pick up their infectious agent that's causing their common cold or their flu virus, and then you touch your eye and it comes through that way. Okay, so those are the different portals of entry. Okay, the next thing we're going to talk about is biosafety. Okay, how can we prevent these microbes from spreading and entering our bodies? Okay, for biosafety, we have both universal or global precautions. So this is things that everybody in the medical and scientific community do. And then we have some standard precautions that have been set by the CDC to protect medical personnel. So first with universal precautions, this is pretty common sense. We treat all blood and body fluids as if they are infectious. This doesn't matter if they are or not, we will treat everything, so all blood and body fluids, as if they're containing disease. Okay, so for our standard precautions that are set by the CDC, we have 8. The first of which is to perform proper hand hygiene. Make sure you're washing your hands before and after entering a patient's room. Okay, 2 is to use your personal protective equipment, your PPE. Do you have your mask on? Do you have your face shield on? Are you wearing your gloves? Are you wearing the little robe thing that the worker is? I forgot what they're called, guys. Okay, the third is you need to maintain respiratory and coughing hygiene. Okay, so if you have to sneeze, you're going to sneeze into your elbow, you're going to cough into your elbow. Okay, you're going to make sure that you're not coughing into your hands that you can then spread to a patient or another human. Okay, number 4 is to use safe injection practices. So if you are using needles, you're going to be careful with them. You're going to make sure they're sterile. They're either going to come directly out of a package. They're not going to be reused by another individual. Okay, you're going to handle sharps safely. This seems like kind of ridiculous to be on this list, but as someone who has to give themselves a shot every week, where I have to drop the fluid into the syringe and then inject myself, you would not believe how many times, especially when uncapping the needle to draw the medicine up, that I accidentally poke my finger or poke the back of my hand with the needle. So it's easier said than done. So this is actually something that they watch quite a bit in the hospital setting in the medical community, because you could be giving a patient an injection, say you're giving them intravenous antibiotics. Okay, you give them that, you pull their IV out, and you accidentally stick yourself with the IV needle. Okay, well, that IV needle has just been in a patient that you know has an infection, and you probably given it to yourself with that accidental needle stick. The amount of accidental needle sticks that happen in the medical community are probably happen more often than we would like to admit. So make sure to handle your sharps safely. Make sure that sharps are placed into the sharps container where they cannot be rescued by people who are looking for needles. Okay, precaution number 6 is that we practice patient isolation precautions. Okay, so different diseases have different precautions. So if you have a patient with a highly infectious and highly virulent disease, they're actually going to be put into special barometric rooms where all the ventilation and the air for that room is controlled and separate from the rest of the hospital, and that air is going to be constantly recycled and goes through HEPA filters to make sure that we don't accidentally transmit an airborne pathogen to everybody else that is in the hospital. Okay, when you are entering a room where a patient is in isolation, you are going to use the appropriate PPE. You're going to make sure you have your gloves on, your mask on, your face shield, your apron, anything that's going to prevent you from transferring the pathogen that is causing disease in that patient to you, and you're going to stop that spread of disease to other patients in the hospital. Okay, number 7 and number 8 also seem to be pretty common sense, but when you are working with a patient, make sure you sterilize the instruments and devices you are using. Okay, we always pull needles out of their sterile pack. If we're using scalpels for surgery, we make sure that they're sterile. They've gone through the autoclave. They come in their own sterile pack. So we just need to make sure that everything's clean and sanitary. And number 8, this one is pretty self-explanatory too. We need to make sure that we are cleaning and disinfecting the surfaces in the environment of those who have these infectious diseases. Okay, so if you're in a hospital room, we're going to make sure we wipe down all of the equipment, at the bed, all the surface areas, the tables, the counters, with bactericide and fungicide and virucide. So cleaning agents that are known to kill those infectious diseases, to make sure that we have a sterile, as sterile as possible environment around the patient. Okay, the next thing we're going to talk about is biosafety. So when we're working with infectious agents that cause disease, there's different levels of biosafety labs that we can use to contain those infectious agents. So we have 4 biosafety levels. Okay, as the number increases, the risk increases in that lab. Okay, so a biosafety level 1 or a level 1 biolab is going to be a low risk lab. Okay, you're not at risk to get a microbial pathogen. You're not going to get any diseases. Okay, this is a level 1 biolab is like our lab there at the school. We're working with non-pathogens. We can just use Clorox wipes to clean up, and there's very low risk of getting a disease from our level 1 biolab. Now, as we go to the level 2 biolab, we're going to start working with pathogens that cause mild disease. So these are going to be pathogens that cause diseases that have a well-documented vaccine and treatment, and both the vaccine and treatment are going to be readily available. Okay, so when we get to a level 2 biolab, we can't just treat it like our classroom lab where we put gloves and goggles on and just use Clorox wipes to clean things. Okay, so this picture on the top left as an example of what's happening here. So she's using a biocontainment cabinet. This is a UV light cabinet. So what's happening is the UV light is keeping the surfaces of this cabinet clean. Okay, this scientist has her PPE on, she has her goggles, she has her gloves, she has her lab coat. Okay, so even though the pathogens she's working with aren't very dangerous, these precautions will catch her from getting the disease even if the treatments are readily available. Okay, so it's a slightly higher risk, but not too worrisome. Now, we start to worry when we get to a level 3 biolab or a biosafety level 3 lab. Okay, because this is when we're going to start working with pathogens that cause serious illness. So we're going to use our PPE like the scientist in this top picture, but we're also going to start working in what is called a biosafety cabinet or BSC. Okay, so this picture here in the bottom right is a schematic of a biosafety cabinet. Okay, so we have our air intake, so the air from the room can either enter from the top or where you're working in the side. Okay, so the air that comes in through the top is actually going to go through some HEPA filters. So it's going to be scrubbed and it's going to be filtered and clean when it comes into the biosafety cabinet. Okay, so you're going to do your work with your pathogen in this cabinet. Unfortunately, it does infect the air. So in this biosafety cabinet, there are air intakes here at the bottom of the cabinet. So the contaminated air is going to go into these intakes, and they're going to travel through the biosafety cabinet and then are going to be run through the couple of filters at the top. And by the time the air exits the top through the ventilation system, the air is going to be filtered and clean, and there is going to be no pathogens found in that air at all. Okay, so biosafety 3 level pathogens, okay, are going to be ones that cause serious disease. So there might be a vaccine, or there might be treatment. There might not be a vaccine, but there's a treatment. Okay, this is our level of risk has increased exponentially from a biosafety level 2 lab. Now, the highest level of biosafety we have is a level 4 biolab. Okay, this is where we work with the highly virulent pathogens like Ebola and hantavirus. Okay, where exposure to either of these pathogens could be lethal for the scientist. So with a biosafety level 3 lab, this is actually going to be a separate room in a building from the level 2 and level 1 biolabs. Okay, so it's going to have its own ventilation system as a precaution, as well as the biosafety cabinet. Now, with a level 4 biolab, those labs are going to be in completely separate facilities. They're going to have their own building on the laboratory campus. Okay, and because they're working with highly virulent and potentially lethal diseases, what the scientists are going to use are the PPE called a full positive pressure suit. Okay, so you can think about this suit as kind of like a space suit that is hooked up to an airline, and it recycles the air in this suit as long as you're in it. Okay, so you have a pump constantly running, pumping air throughout your suit, you're keeping the air free of pathogens. You are going to be wearing several layers of gloves, and where your suit meets your gloves, you're going to tape that part. You're going to tape where the bottom of the suit meets your shoes. You're going to wear special boots. Some of the positive pressure suits are going to be a little bit like onesie pajamas, where the boots are going to be attached to the suit. Okay, so biosafety level 4 bio sorry guys, biosafety level 4 labs are going to be the highest risk labs, especially because a lot of these don't have vaccines and they don't have treatments. Okay, so catching these pathogens could be a death sentence for what we are studying. Okay, so think Ebola. We have to study Ebola in a level 4 biolab. Okay, so that is section 2.4. We talked about how diseases can spread and the possible portals of entry. We talked about the various precautions that you can have for handling infectious materials, and we talked about the 4 biosafety levels in the lab. Okay, so we are actually going to go on to 2.5. But before we go on to 2.5, let's do review question number 3. Okay, so determine the biosafety level of the following pathogen. Okay, so Agent A can cause severe disease, including death, in some patients. It can cause disease by either inhalation or ingestion. It can be treated with the readily available antibiotic doxycycline. Okay, so is this going to require a biosafety level 1, biosafety level 2, biosafety level 3, or a biosafety level 4? So pause the video and take a second and try to figure out what the answer to this question is. Okay, so let's look at what we know from the problem. So we know that Agent A can cause severe disease and death. So this is going to eliminate A and B, the biosafety level 1 and the biosafety level 2 labs. Okay, so we have to choose between whether it's a level 3 or level 4 lab. Now, we really don't care about how we can get the disease, but what's a really important piece of information is that it can be treated with the antibiotic doxycycline. Okay, so this tells us that it is not D, it's not a biosafety level 4 lab where there's no treatments. So the answer to this question would be C, the biosafety level 3. Okay, so section 2.5 and 2.6 are pretty short, so we're just going to append them to the 2.4 video. Okay, so 2.5 is all about the factors in the host that can lead to disease. So the objectives for this section are going to be that you're going to be able to define different biological features in human hosts that can influence the host throughout a course of an infection. Okay, and the second is that you're going to be able to explain how a host's behavior can impact or increase their susceptibility to disease. Okay, so what we don't talk about a lot until we get into microbiology is that there's different health and lifestyle factors that are going to contribute to whether you get a disease or not. Okay, the first factor is going to be age. Okay, so children underneath the age of 3 and the elderly that are over 60, they're going to have the greatest susceptibility, so they're going to be more likely to catch a disease. Part of this is because they're on the opposite ends of the spectrum. So children underneath the age of 3, they're barely gaining their immune system, it's barely ramping up, so it's very fragile. So they're going to be susceptible to disease. And the opposite happens with the elderly. They're getting towards the end of their lives, their immune system's not working as great, so they're going to be more susceptible to disease because of it. Okay, another factor that can lead to catching a disease is your literal genetic makeup. So, what kind of receptors do you have on the outside of your cells? Okay, so those who have a genetic makeup that makes them more susceptible, there's going to be more receptors on the outside of their cells through the various tissues that where bacteria and viruses can bind and can trick your cells into taking them into the cell and infecting them. Okay. Another factor is this is a pretty commonplace one is the host hygiene and behavior. Are they good? Is the host good at washing their hands? Do they use hand sanitizer? Do they make sure their food is cooked properly? Okay, are they using protection when having sex? Okay, so host hygiene, the better the hygiene, the less susceptible you are going to be to disease. Okay, and another factor that we have to take into account, and this one's been getting a lot more study over the last 10 years or so, and that is how healthy are you? What is your nutrition level? Are you eating a bunch of fast food or are you having vegetables and well cooked meals? Okay, and the amount of exercise can actually affect whether you can catch a disease or not. So, having good nutrition and doing moderate exercise 3 times a week for 30 minutes will actually boost your immune system and make it so that you don't get sick as often. Okay, so other factors that can lead to you getting an infectious disease is pre-existing conditions. Okay, do you have an autoimmune disease like lupus or type 1 diabetes? Do you have a chronic infection? Do you have cancer? Okay, do you have a suppressed immune system? Okay, do you have innate or acquired immunosuppression? Okay, pre-existing conditions, sometimes like heart disease, will help pave the way for those microbes to infect you and give you the disease. Okay, another big lifestyle factor would be your occupation. What is your job? Okay, so healthcare workers, laboratory personnel, farmers, agricultural workers, hunters, sex workers are all at higher risks of exposure. Okay, healthcare workers, obviously, because they are working with those that have the disease. Laboratory personnel, they might be studying the pathogen. Farmers and hunters, they're out where the reservoirs for these diseases could be. Okay, hunters are in the woods, they're exposed to ticks, they're exposed to mosquitoes, so they can possibly pick up a pathogen through an insect vector. Agricultural workers, they could just by tilling the land, they might stir up some pathogens, right? We have our Bacillus anthracis that causes anthrax that mostly lives in the soil. Now, sex workers, this is not one that you typically think of in high school, but sex workers, this is a very dangerous occupation for getting infectious disease because there's a lot of direct contact in different points between the sex workers and their clients. Okay, so your immune status and immunopathogenesis. Okay, so how your immune system fights disease. A lot of people are immunocompromised. Okay, and because they're immunocompromised, their immune system can actually cause more damage in the healthy tissues, which lead to an increased susceptibility to infectious disease and getting those diseases. Okay, that is 2.5. Make sure you know some of these factors and are able to explain them for the exam. Okay, so this is going to lead us into the final section of chapter 2. This is 2.6. 2.6 deals with global change and emerging infectious disease. So the objectives for this section are you're going to be able to explain how civilization has impacted emerging infectious diseases, and you're going to explain how climate change can alter infectious disease patterns. Okay, so we need to learn some vocabulary. So the question that this slide asks, are all infectious diseases known to us? And the answer is no. Most infectious diseases, especially new ones, can be categorized as either an emerging disease or a re-emerging disease. So an emerging disease is one that has recently appeared in the population, so we haven't seen it before. So COVID-19 would be an emerging disease. We had never seen it before the pandemic happened. A re-emerging disease is a disease that is known to us, who we've used vaccination campaigns in order to mostly eradicate those diseases. Okay, so a re-emerging disease is when these diseases who have gone away come back. So an example of this is polio and the measles. Because of anti-vaccine campaigns, some parents aren't vaccinating their children, and because of it, we're starting to see more and more measles outbreaks throughout the world. Okay, one of the most significant ones that we've seen in recent years happened at Disneyland, where a homeschool anti-vaccine family went to Disneyland. Their child was carrying the measles and passed it on to the other children in Disneyland that didn't have those vaccinations or who were immunocompromised. So there was a small outbreak of the measles just by going to the happiest place on earth. Okay, so with emerging diseases, the way that these become known to us are usually because of changes in the environment around them. So we're seeing a lot more increases worldwide of death and disease that are connected to human activities. Okay, aka we're doing things that are bringing us closer to disease reservoirs and disease vectors that we haven't come into contact before, and haven't come into contact with before. Okay, which is going to bring us to the last thing I want to talk about. Okay, is how are we coming in contact to these emerging diseases? So one of it would be urban sprawl. So our urban areas are getting larger and larger. So we're taking over the natural land. So we're starting to cut down woods and forests. So and we're building on them. So urban sprawl is bringing us into contact with these disease reservoirs that we normally wouldn't have been in contact with because we are building where they are. Okay, deforestation is a huge one here because we're cutting down the ecosystems and the environments that these disease reservoirs, that can be an animal reservoir, we're cutting down their homes. So now we're coming in contact with these animal reservoirs and we're starting to see the emergence of some of these diseases where we normally wouldn't have come in contact with those animal reservoirs before. So climate change is also one that we're seeing a big effect on disease because the climate is getting warmer and warmer and warmer. we're seeing a lot of infectious disease not going away. Because one of the things that winter helps with some diseases, especially ones that cause disease in humans, is that they like specific temperatures. They grow well in warm, so around that 37 degrees Celsius or 97.3 degrees Fahrenheit temperature range. Okay, so when it gets cold, those that grow great at those temperatures are going to die off and disappear. But because global warming and climate change is happening, the temperatures are increasing, which means that we're not getting as long of cold spells. So some of these diseases aren't dying down like they used to. So they're going pretty much all year round now. Okay. Other things are like natural disasters, so drought and excessive rain can also bring animal reservoirs and other vectors into contact with humans and help bring these new emerging diseases to humans. So this is a really great summary to read through because you are going to need to know for the exam different and for your homework different ways that humans are coming in contact with new diseases. Okay, a lot of the emerging diseases that we're seeing are usually zoonotic diseases. So diseases like COVID-19, where we think they have jumped from an animal host to a human host and are now causing disease in humans and not just those animals. Okay, so this concludes section or chapter 2. We're going to go over chapter 3 next, and chapter 3 is the last chapter we have before the unit 1 exam.